Hello everybody and welcome to another video. I'm amazingly anatomical and I'm so excited to make videos on the human body and science in general, and as usual, all from a high school perspective. Today I'm going to continue exploring the brain, but I'm going to go more into the chemistry of the brain and how drugs affect the brain. Let's get started. The chemistry of the brain. Communication of information between neurons is accomplished by the movement of chemicals across small gaps called synapses. Chemicals called neurotransmitters are released from one neuron at the presynaptic nerve terminals. The neurotransmitters cross the synapse where they can be accepted by the neuron at a specialized site called a receptor. For example, acetylcholine is a neurotransmitter that slows down the heart after the electric stimulation of the vagus nerve. For a chemical to be a neurotransmitter, certain criteria have to be met. The chemical must be produced within a neuron. When the neuron is stimulated, a neuron must release the chemical. After a chemical is released, it must be inactivated. The inactivation can be through a reuptake mechanism by an enzyme that stops the action of the chemical. When the chemical is released, it must act on a postsynaptic receptor and cause a biological effect. If the chemical is applied on the postsynaptic membrane, it should have the same effect as when it is released by a neuron. While these are the criteria that classify a neurotransmitter, it is important to note that not all neurotransmitters actually meet all the criteria. Many types of chemicals act as neurotransmitter substances, such as small molecule transmitter substances like histamine and epinephrine, amino acids like aspartate, gamma-aminobutyric acid, and glycine glutamate, neuroactive peptides such as insulin, glucogen, dynorphin, neurotensin, and motilin, and soluble gases like nitric oxide and carbon monoxide. I'll go more into these chemicals in a separate video. Neurotransmitters are made in the cell body of the neuron and then transported down the Amazon to the Amazon terminal. Molecules of neurotransmitters are stored in small packages called vesicles. Neurotransmitters are released from the axon terminal when their vesicles fuse with the membrane of the axon terminal, spilling the neurotransmitter into the synaptic cleft. Nitric oxide, however, is not stored in synaptic vesicles. It is released soon after it is produced and diffuses out of the neuron. Then it enters another cell where it activates enzymes to produce second messengers. Neurotransmitters will only bind to specific receptors on the postsynaptic membrane that notice them. Neurotransmitters can be stopped by 4 different mechanisms. It can be stopped through diffusion, where the neurotransmitter rifts away out of the synaptic cleft where it can no longer act on a receptor. Enzymatic degradation or deactivation is when a specific enzyme changes the structure of the neurotransmitter so it is not recognized by the receptor. Astrocytes, a type of glial cell, can also remove neurotransmitters from the synaptic cleft. The final way, reuptake, is when the whole neurotransmitter molecule is taken back into the axon terminal that released it. This is a really common way for stopping the action of norepinephrine, dopamine, and serotonin, where they can no longer bind to the receptors after getting removed from the synaptic cleft. Now I'm going to go into how drugs alter the brain. Drugs can alter with the way neurons send, receive, and process signals via transmitters. Drugs like marijuana and heroin can activate neurons due to the fact that their chemical structure mimics that of a natural neurotransmitter. As a result, this allows the drugs to attach onto and activate the neurons. The big problem with these drugs is that they don't activate neurons in the same way as natural neurotransmitters do, and lead to abnormal messages being sent through the network. Amphetamine and cocaine can cause the neurons to release abnormal amounts of natural neurotransmitters or prevent the normal recycling of these brain chemicals by interfering with transporters. Like with marijuana and heroin, cocaine and amphetamine amplifies and disrupts the normal communication between neurons. Additionally, the basal ganglia, the extended amygdala, and prefrontal cortex are all affected by the intake of drugs. Going back to the impact of oxygen from my last video, chemically, a lack of oxygen also significantly decreases the levels of serotonin, which is a critical neurotransmitter that modulates mood and anxiety. As a result, people can become depressed or have anxiety with low levels of oxygen. Low levels of oxygen also increases the level of dopamine, which is the neurotransmitter responsible for regulating pleasure. This sort of balances the lack of serotonin. Furthermore, a lack of oxygen can worsen the symptoms of people with bipolar disorder with anxiety. And that wraps it up for this video. Thank you guys so much for watching and listening. Please make sure to check out my previous video and stay tuned for more videos to come. Bye.